team, welcome back to the 2.5 Genetic Variation and Change 4-credit external video series. This is video 7. In this video, you'll be learning about the 3 genetic inheritance patterns of complete dominance, incomplete dominance, and codominance. And by the end of this video, you should be able to distinguish between complete dominance, incomplete dominance, and codominance, and you should be able to solve the genotype and phenotypes of individuals from crosses involving these types of inheritance patterns. So first of all, let's recap from our previous lessons. I've taught you that in genetics, all traits or characteristics are controlled by genes, and different versions of a gene are called alleles. All organisms have 2 alleles for a gene. One allele has been inherited from one parent, and the second allele is inherited from the other parent. And the way those 2 alleles that you've inherited from your parents, the way these 2 alleles interact, determines the organism's phenotype or their observable traits. So for example, the trait for solid dark coat colors in cats is controlled by the brown gene located on the B locus of the cat's genome. This brown gene has 2 alleles, or 2 genetic variants. One allele is the black wild type allele, capital B, and the other allele is the chocolate allele, little b. A cat inherits 2 alleles for this gene from their parents, and the way those 2 alleles interact determines the organism's phenotype, so whether the cat has a coat color that's black or that's chocolate. So just to be clear, a homozygous genotype will always produce a phenotype that matches the allele, regardless of the inheritance pattern of the gene in question. So if the big B allele is a black coat color variant, then the homozygous genotype of big B will produce a black coat color phenotype. And same goes with the little b allele. So if the little b allele is a chocolate coat color variant, then the homozygous genotype of little b will produce a chocolate coat-colored phenotype. It's when the genotype is heterozygous that the phenotype will differ depending on the inheritance pattern of that gene. So there's either complete dominance as an inheritance pattern, incomplete dominance, or codominance. And I'll now explain each of those 3 inheritance patterns further. Let's start off with complete dominance. In this pattern of inheritance, one allele completely masks the other allele. In the heterozygous genotype, the dominant allele is expressed while the recessive allele is hidden from the phenotype. So for example, for the trait that I mentioned before, the solid dark coat colors in cats, this is controlled by the browning gene. It has a complete dominance inheritance pattern. So the big B allele is black and is dominant and completely masks the chocolate little b allele, which is recessive. So the heterozygote will always express the dominant allele. When these 2 heterozygotes are crossed, so when a big B little b is crossed with a big B little b individual, the genotype ratio is 1 homozygous offspring to 2 heterozygous offspring to 1 homozygous recessive offspring. So 1 big B, 2 big B, and 1 little b. The phenotype ratio is 3 black offspring to 1 chocolate offspring. Notice how there are only 2 phenotypes compared to the 3 genotypes there are. And that's because the homozygous dominant and the heterozygous offspring all present as the dominant black phenotype because the big B allele is dominant and will mask the little b allele, which is recessive. In another example, the trait of plant height is controlled by a gene that has a complete dominance inheritance pattern. The tall big T allele is dominant and completely masks the short little t allele, which is recessive. And when we cross 2 purebred plants together, so a purebred tall and a purebred short, what we get is that all offspring have a heterozygous genotype and all the offspring are tall because the big T allele masks the short little t allele. Now let's have a look at the second pattern of inheritance, incomplete dominance. In this pattern of inheritance, neither allele is completely dominant over the other allele, hence incomplete dominance. When both alleles are present in the heterozygous genotype, neither alleles are fully expressed. Instead, the heterozygote has a phenotype that's a blend of the two alleles. For example, let's look at the trait of snapdragon flower color, which is controlled by the color gene, C, and has an incomplete dominance pattern of inheritance. Neither the red flower, CR, nor the white flower, CW, is completely dominant over the other allele. And when both CR and CW alleles are present in the heterozygous genotype, neither allele is fully expressed. Instead, the heterozygous has a phenotype that is a blend of the two alleles. We don't see a red or a white phenotype, we see pink. And pink is an intermediate or a blend of the red and white genotypes. So when two heterozygotes are crossed, so a CR-CW is crossed with a CR-CW, or a pink is crossed with a pink plant, the genotype ratio is one homozygous CR-CR offspring to two heterozygous CR-CW offspring to one homozygous CW-CW offspring. The phenotype ratio is one red plant to two pink plants to one white plant. And notice that now there are three phenotypes available or present compared to the two phenotypes that were seen in complete dominance before. This is because incomplete dominance inheritance produces a new intermediate phenotype for the heterozygote. The heterozygotes are always intermediate. They're not exactly like one of the parents, they're a blend of both parents. Here's another example. This is the trait for color point restriction in cats, and it's controlled by the C gene and also shows an incomplete dominance pattern of inheritance. So for your information, color point restriction is a trait where more pigment can be found on the cooler parts or the end points of the body, such as the feet, the face, the ears, the tip of the tail, and sometimes the back. In this scenario, there are two alleles for color point restriction. There's the Burmese color point restriction allele, CB, and the Siamese color point restriction allele, CS. A homozygous CB-CB cat will express the Burmese color point restriction phenotype. And this is where the body is only slightly lighter than the extremities. A homozygous CS cat will express the Siamese color point restriction phenotype, where the body is really pale and the pigment is restricted to only the extremities. But when both CB and CS alleles are present in the heterozygous genotype, neither the Burmese or the Siamese color point restriction alleles are fully expressed. Instead, this heterozygote has a phenotype that is a blend of the two alleles, and a new phenotype emerges called mink color restriction. The mink phenotype is an intermediate between the Burmese and the Siamese. In the mink color restriction phenotype, the body is not as dark as the Burmese and is not as light as the Siamese. It is somewhere in between. Now let's look at the third and final pattern of inheritance for this video, codominance. The prefix co means together, like cooperate means to work together, and coexist means to exist together. In this inheritance pattern, both alleles are expressed together, equally, and independently in the phenotype. Instead of blending, like what we see in incomplete dominance, the heterozygote shows both traits in this phenotype. For example, the trait of cattle coat color has a codominance pattern of inheritance. In the heterozygote, both red R and white W alleles are expressed together, equally, and independently in the phenotype. And this results in a cattle coat color with distinct patches of red and white hairs rather than a blend of color. In another example, the trait of feather color also shows a codominance pattern of inheritance. In the heterozygote, both white W and black B alleles are expressed together, equally, and independently in the phenotype. This results in speckled feathers that are both white and black, not grey feathers. Here's a human example of codominance in human blood types. Human red blood cells have these things called antigens presenting on the surface of our red blood cells. And these are protein markers that signal to other cells in our bodies, like white blood cells. In the human ABO blood group system, the IA and IB alleles are codominant to each other. So in the heterozygote, both A antigen, IA, and B antigen, IB, are expressed together equally and independently in the phenotype. So the result of codominance is red blood cells presenting both A and B antigens on its surface, producing type AB blood. You might be wondering, why is the letter I used to note the alleles in the ABO blood group system? Great question. The letter I is used because it used to stand for isoagglutinogen. Now we just use the word antigen. So whenever you see I in the context of blood, just think that means antigen. So to summarize, an individual inherits 2 alleles from their parents, 1 allele from their mum and 1 allele from their dad. And the way those alleles interact with each other determines the organism's phenotype. Homozygous genotypes will always produce a phenotype that matches the allele. It's when the genotype is heterozygous that the phenotype will differ depending on the inheritance pattern of that gene. In complete dominance, the dominant allele masks the recessive allele. In incomplete dominance, neither allele is dominant over the other. The phenotype is instead a blend of the 2 alleles. And in codominance, both alleles are expressed together equally and independently in the phenotype. So by now, you should be able to distinguish the difference between complete dominance, incomplete dominance, and codominance. And you should also be able to solve the genotype and phenotype of individuals from crosses involving these 3 inheritance patterns. Thanks for watching, and I'll see you in the next video.